Good evening, everyone. Once again, welcome to the second day of our online Bioinformatics Foundation workshop. This is Leah Simon, faculty at JaneFit Hub, and I'm glad to have you all back for this evening session. Today, we will focus on sequence formats, sequence alignment with some basic interesting exercises. This will be the workflow of today's session, beginning with the fundamentals of sequence formats, leading into alignment methods and tools, and concluded with real-world applications and phylogenetic analysis using Mega software. Before explaining sequence formats, it's important to understand what kind of data which is used in bioinformatics. Now you are all familiar with the different databases from yesterday's session. Now we are using some sequenced data of nucleotide or proteins. It is a long string of letters. For DNA and RNA, these letters are nucleotide bases. A for adenine, T for thymine, C for cytosine, G for guanine, and U for uracil, which is present in RNA. And in case of protein, the data is a sequence of amino acids represented by single letter codes like M for methionine, L for leucine, etc. Here, nucleotides and proteins itself is insightful data. Then why we need sequence formatting? Here is your answer for you. I'm sure that some of you recognize the people, but it's not our concern now. Look at this image. We can see 7 people dressed casually, each in different poses and in messy looks. Now look at the second one. The same people. Did you notice any change? Their eyes are focused, their standing position, body posture are uniform and formal. What is the change here? It's a uniform. I think this is a good analogy to understanding the difference between common sequence data and formatted sequence data. So here is the answer why we need sequence formats. In bioinformatics, handling raw or common sequence data without a structure or a uniform would be inefficient because it is hard to interpret without a context. That's where sequence formats come in. So let's go for what is sequence format. A sequence format, it is a structured way of organizing biological sequence data, which are DNA, RNA, or protein, to make it easier to read and analyze. This is one of the example for common sequence format. Here you can see that there is a black color image which contain a header line starting with a greater than symbol and a second line with some nucleotide bases. This is an example for sequence file format. Here the header line starts with a greater than symbol. That means it is referred as a identifier and followed by a brief description of the sequence. It may be its name, ID, source, etc. And in the second line, it will be the data, which may be nucleotide protein or any other relevant biological sequences. So this is the a common format of file formats. And also, there are several file formats used in bioinformatics. Some of them are FASTA, FASTQ, BCF, SAMBAM, etc. And some of images of FASTA formats are here now. In this session, we are only handle the FASTA and FASTQ formats. Here, FASTQ is a key format used in high throughput sequencing like next generation sequencing, containing both nucleotide sequences and quality score for error detection. While widely applied in advanced sequencing workflows only. In this session, we are mainly focused on FASTA format for our hands-on exercises. Okay, now from this table, we can understand that what changes happen to a common data when it is formatted and the difference between FASTA and FASTQ. Here we have two types of data, nucleotide and amino acid, that means protein. And after that, we can see that there is two kinds of format here, FASTA and FASTQ. In case of nucleotide, the FASTA format looks like this. That means we already know that there is a header line for a format. So in FASTA format, the header line starts with a greater than symbol. It is called identifier. Along with the greater than symbol, there is a description. First one is an ID, then the organism and the type of data. Here it is a insulin mRNA data of Homo sapiens. This is what FASTA format. And in case of protein, it's also the same. The only difference is it's a protein data. So after the greater than symbol, there is a description about that protein sequence and also which organism takes. And in case of FASTQ, it's starting with @ symbol. The first line, the header line starts with the @ symbol. Along with the symbol, after a space, there is a sequence description. And the second line, there is a sequences, which may be nucleotide and also the protein. And in the third line, it is a plus sign. And in the fourth line, they give a quality score. This quality score helps us to assess the sequence-based quality. Try to understand that this is a format accepted by some tools or databases of bioinformatics. So just understand that FASTQ is a 4-line format, and it starts with @ symbol with a description, then the data, then a plus sign, then a quality score. It's just a format. Okay, this is what FASTA and FASTQ. Here is an example for FASTA and FASTQ format images. From this, you can see how FASTA looks in databases. Here it is. And in case of FASTQ, it starts with the header line that contains an identifier. There is also a description on the same line. Don't confuse with this sequence itself. If there is no space after the identifier, in case of FASTQ format, you can see that here there are 4 lines here, and they labeled each line what each line contains. Okay, in the first line, the identifier is @ symbol. After the @ symbol, there is no space here. So if there is a space, then the next string will be the sequence description. Here there is no space after the identifier. So this line is also considered as identifier. Okay. In the next line, or in the second line, they give the sequence data. And in the next line, there's a plus sign. Along with the plus sign, the same identifier which we already seen in the first line. Okay. In the last line, there is a quality score. This is all about the FASTQ format. Okay. Now we are understand what is sequence formats and the basic sequence format we are used in bioinformatics. So we can go to the sequence alignment. Sequence alignment is the process of comparing 2 or more biological sequences. Like the image, we can compare human and chimpanzee. It helps to identify regions of similarity. These similarities helps us understand functional, structural, or evolutionary relationship between sequences. Sequence alignment is a foundational technique in bioinformatics analysis. Okay, and there are mainly 2 types of sequence alignment. That means they are divided into 2 categories: global versus local alignment and pairwise versus multiple alignment. Here, global versus local alignment focus on the extent or length of the alignment. I will explain it later. And the pairwise versus multiple alignment focus on the number of sequences being compared. Okay, so going to the global versus local alignment. From this table, you guys can see that there are the details about and the differences between local and global alignment. In case of global alignment, the name indicates that it is done globally or fully across the sequence. One suggestion for global alignment is that the sequence length should be similar because it is suitable for comparing similar sequences. In case of local alignment, which perform locally, as indicated by its name, or partially across the sequences. It is suitable for sequences of different length and is best for comparing sequences that have regions of similarity but also overall differences. Okay. And here there are some other details are given in this table. Gap penalties, I will explain about it. Gaps are distributed across the entire sequence in global alignment. But in case of local alignment, gaps are only introduced if it is necessary. And the output will be because the global alignment aligns from the start to the end. So the output will be like that. And in case of local alignment, the aligns only regions with high similarity. Okay, and the main use of global alignment is genome-wide comparison and evolutionary studies, and some other applications are there. And in case of local alignment, there are some finding which helps to finding conserved domains and motif detection. And some algorithms are works behind these alignments. In case of global alignment, the algorithm is Needleman-Wunsch algorithm, and in case of local alignment, it is Smith-Waterman algorithm. Okay. I will explain this with an example. Imagine that, like this image, you are downloading 3 genome sequences from NCBI. Okay, one is for human, other is for chimpanzee, and one is for coconut. We would perform a global alignment between human and chimpanzee sequences and local alignment between the human and coconut sequences. First of all, we need to download the sequences from NCBI. Then we need to align the downloaded sequences globally and locally. So, okay, now we align the sequences globally and locally. In the global alignment, you guys can see these 2 images here. I already labeled the target sequence is human in both local and global alignment, and the query sequence is chimpanzee in global alignment. Okay. In case of global alignment, our target sequence is human sequence and the query is the chimpanzee. We align the entire sequence from start to end. We can see here, from here to here. We are aligning the both sequences. And the similar or matched nucleotides are connected with the lines like this. You can see here. There are lines in between target sequence and query sequence, which are the similar nucleotides in the present in the target and query sequences is labeled with a line. You might also notice insertions in the query sequence is called. You can see that there is a minus-like structure here. That is called insertions. It is called gaps. A gap is it is a space added to help match one sequence with another, especially when their lengths are slightly different. So we can do that if there is any length difference between target and query sequence is also okay in global alignment. At that time they insert a gap to get maximum similarity between the target and query sequence. Okay, this is what global alignment done. Okay, we have done our global alignment between human and chimpanzee genome. Now, in case of local alignment, our target sequence is a human genome. Here the target sequence is a human genome and the query is a coconut genome. What do you think about the similarity between the coconut and the human sequences? While humans and coconuts are very different, there can still be some relationship between the genomes. However, this doesn't mean that coconut gradually evolves into human. It means local alignment helps us to find small regions of similarity between genomes, even for very distantly related organisms. So here we can see some similarities between human and coconut genome. So for example, certain genes involved in cell division or metabolism may be similar because they perform the same basic function in both plants and animals. Local alignment focuses on finding these shared regions like this, showing how different species can still be connected at a genetic level. Okay, this is what global alignment and local alignment. And we can summarize it that global alignment compares sequences across their entire length, making it ideal for closely related sequences like human and chimpanzee. And the local alignment, on the other hand, focuses on finding regions of similarity within sequences, even if the sequences are very different, like between the human and coconut. Both methods help identify important genetic relationships either across full sequences or specific regions. You can see from this graph too. Okay, so we are completing the global and local alignment. Now that we know about the matches and mismatches and also the gaps also. So let's move on to the next type of alignment, pairwise sequence alignment and the multiple sequence alignment. Compared to the global and local alignment, this is very easy to understand. The pairwise sequence alignment only compares two sequences. Here, the concern is the number of sequences. So pairwise sequence alignment compare only two sequences. You can see an image here. Here, the first sequence compare with the second sequence. The first sequence, there is an adenine in the first place, and also in the second sequence also have an adenine in the first place. So we can understand that there is a match. And in the second place, there is a cytosine in first sequence and thymine in the second sequence. So it is a mismatch. And this is what they analyze from the beginning to the end of the sequence. At the end, we can see a gap. We know that why they insert a gap here to get the maximum similarity. So this is what done in pairwise sequence alignment. In case of multiple sequence alignment is also understand by this image. We are compare more than two sequences, and in this image it is easy to understand the similarities because they give different colors for each nucleotide bases. So we can easily find out where the adenine is because in case of the first residue, we can see that sequence A to sequence F have a similarity, and also the G is get a full green line here. So from the colors, we can easily understand how much these sequences are similar. So multiple sequence alignment is used to compare more than two sequences. Okay. And in this session, we are also handling some tools which we used for pairwise and multiple sequence alignment. In case of pairwise sequence alignment, we are using mainly the two, we have some other tools are there. In this session, we are handling BLAST and EMBOSS. BLAST stands for Basic Local Alignment Search Tool. So already we can understand that it is a local alignment search tool. And EMBOSS stands for European Molecular Biology Open Software Suite. It's for global alignment. Okay. BLAST is a fast tool that searches large databases for similar sequences. Make it great for finding matches quickly. But EMBOSS offers a suite of tools which provide more detailed alignment but take longer to process. Okay. We will use BLAST for a quick demo here. It is a search engine of NCBI that finds similar sequences from vast databases. This shows how easy it is to compare our sequences with other to find matches or study the relationship. Okay, if you have your system or mobile phone in with you, you can search in your browser. Go to the browser and search for NCBI. From there, you can find BLAST. Here is the BLAST interface, and we can see that there are different types of BLAST here. The first one is nucleotide BLAST. Nucleotide BLAST is for nucleotide against nucleotide database. And the protein BLAST is for protein against protein database. And there are other options are here, BLAST-X and T-BLAST-N. BLAST-X is for translated nucleotide against protein database. And T-BLAST-N is for protein against translated nucleotide database. Here I choose protein BLAST because protein sequences are shorter and more manageable to do sequence alignment compared to the nucleotide sequences. making the comparison faster and easier for this demo. Okay, so we can click on the Protein Blast. Here in this protein suite, you can see one more option here. In the interface, we have only 4 options, and in here we can see another option is T-BLASTX. T-BLASTX stands for translated nucleotide bases, translated nucleotide database. Okay. Here you can see there is a query space here. Here you can paste your accession number of your data or the FASTA sequence. And also there is another option is here. You can choose your file also. If you paste or copy paste your data from any sequence databases in FASTA format to a text file, you can choose that file here also. Now I'm going to paste my data here. I already have a hemoglobin FASTA file data. So copied the human hemoglobin FASTA data and paste it here. And here are some other parameters are here. I never going to change anything here because it is a demo session. If you want to change for your need, you can. And this is enough for to do find out the best sequence alignment using BLAST. Okay, so click on the BLAST. It takes some time based on your data. Okay, now we get the resulted window. From the result, we can see that there is a basic details about the job we are already done. Here you can see that your data name here, it is human HB. You can change it if you need it. And there are some other details are here. Description of the data, molecular type is amino acid, and the query length is 105. This is the basic details about our query data. And in the end, we can see that there is 4 sections in the description in the resulting window. The first one is description. The second one is graphic summary and alignments and also the taxonomy. In case of description, here I choose. Here you can see that here you can download your alignment by using this drop down menu. And here are some other here some formats are here. If you want this as for your study, you can download it from here. And also you can select which column you needed in your result. I only need the description, scientific name, and query coverage, value, and percentage identity. So, okay, this is what we need in this session. So you can choose which column you needed here, and also have an option to get how many alignments you needed. You can go to 10, 50, and 100. Okay, here we choose 100. And in the description, we can see that there is a description of the data and the scientific name of the organism which possesses the similar aligned sequences of our query data. And here the query coverage shows the percentage of your sequence that align with the matching sequences. So here we can see that most of the sequences shows 100% query coverage. And the next one is e-value. E-value indicates how likely it is that the match occurred by chance. The E-value is mainly the value between 0 to 1, and how less the E-value, the data will be that much significant. That means it gives that if the E-value is less, the alignment similarity will be high. Okay. In the percent identity, we can see that the identity of insulin, our query data with the alignment will be from here. It is also it also shows the similarity of your sequence to the matched sequences, which expressed in percentage. This is what we need for this session. So this is what is description. And in the graphic summary, you can see the aligned sequences in a coloring lines. Here the red line shows that our alignments are very much similar to our query sequence. So you can see that there is an alignment score based on the color. If your data is not that much similar, you can see in pink color. If it is if the similarity between 80 to 200, we can see it in pink color. Or if it is lesser than that, it will be in green, blue, or in black. This is what the graphic summary. And you can also touch your cursor into the each line. You can see the which organism possesses that insulin sequence. Okay, this is what graphic summary. And also you get the alignments from here in the alignment option. The full alignment will be here. Still loading. And at the end, there is a taxonomy option. From here, you can understand the lineage, organism taxonomy, and other details from the taxonomy option. If you are going for a taxonomy study or phylogenetic analysis, it will be helpful for you. Now, we need only the description area. Okay, this is what BLAST result. The next one is multiple sequence alignment. We already know that what is multiple sequence alignment. In multiple sequence alignment, we can align more than 2 sequences at a time, and it helps to find out better predictions of phylogenetic analysis. And here I choose a data of hemoglobin from different organism. We can easily understand that how these or the hemoglobin in these organisms or the protein in these organisms are related by performing multiple sequence analysis. In multiple sequence analysis, we can have different tools: Clustal Omega, Muscle, Marked, Clustal W, and so on. There are some other tools are there. Here we will go to the Clustal Omega and Muscle. Okay, Clustal Omega and Muscle is almost similar type of interface. In case of Clustal Omega, this is the interface of Clustal Omega. You can easily search it through your browser to Clustal Omega, and you can go. You can be here in this interface. In Clustal Omega, you can choose your file from this option, choose file option. Here our input file is a hemoglobin data from different 5 organisms. Here you can paste it here all the sequence, or you can choose file here and submit it. You will get a result. The resulting window looks like this. Okay, this is the Clustal Omega resulting window. Here in this image, you can see that the 5 sequences are aligned here, and the similarity is represented in star, and the dissimilarities are in colon and in dot. Okay, and also you can see it in a coloring format too. This is what Clustal Omega. And in case of Muscle stands for multiple sequence comparison by log expectation. Okay, we will do it. Go to your browser and search for Muscle. Okay, this is the muscles interface. It's also same as the ClustalW. You can choose your file here. I already have a text file with the 5 sequence data. So I'm choosing this file and open it. Go to the end and submit it here. Okay, you can view your results. This is what your result is. Here we can see that there is not much similarity between these protein sequences from the 5 different organisms, because we know that there are entirely different organisms: butterfly, cat, human, elephant, and snake. So this is your multiple sequence alignment in muscle, and you can also view this with a color alignment here. Okay, and from muscle only you can get other alignment forms and also a phylogenetic tree. I will explain it in the next session. And you can download your files from here. You can download it in different formats and based on your need. Okay. This is what multiple sequence alignment using muscle. Okay, now we are familiar with the muscles result also. So now we can go to the applications of sequence alignment. Here highlights the applications of sequence alignment, which is very crucial in various fields of bioinformatics. First, it is used for phylogenetic analysis, helping us to understand evolutionary relationships. Additionally, sequence alignment aids in identifying homologous genes, which can reveal similarities between different species. Okay, one key application is constructing a phylogenetic tree. We will do in the next session. Visual representations that depict the evolutionary pathway of organisms. Understanding this relationship is vital for studying biodiversity, evolution, and the development of targeted treatments in medicine. Therefore, focusing on phylogenetic tree analysis is important as it provides insight into the evolutionary history and functional relationships among genes and species. Before going to the software, we need to understand what is a phylogenetic tree. Here you can see 2 images of phylogenetic tree. This is just a model of phylogenetic tree, and it is a rooted tree. There are different types of trees which can made by the softwares you are using. Here we are using the Mega software. In Mega, you will get a output like this. Here is the image of phylogenetic tree. You can see that here is a line which is called root. This root indicates the common ancestor of all the species in this tree. Okay. And the root ended in a point called node. The node is act as the common ancestor of the branches which starts from the node. Okay. These lines starts from the node is called branches. And again, there is a stopping point. Then again, there is a branch started from it. So from the root, there are some branches, and at the end, we can see a labeled area A, B, C called leaves. Branches are the evolutionary path taken by the different species from the common ancestor, from the root. And leaves are the end point of the branches, which represents the current species, such as humans, chimpanzee, or mouse. Okay. And the second diagram, we can easily understand that how the species evolved from their common ancestor and diverged into separate lineages over time. Maybe this in the first session, first common ancestor is very much different with this end species one. Okay, so this is the difference between the two trees, and I think you guys will get a basic idea about what a tree and what is the representation in the tree. Okay, so we can summarize that the tree helps us to understand how these species are related and how they evolved from a common ancestor. So now you can go to the very interesting session and the last part of this seminar is Mega software. Mega is one of the powerful software which we used to conduct molecular evolutionary analysis. Here we use Mega 11. There are some other versions are there. It provides features for constructing phylogenetic trees, conducting sequence alignment, and also estimating evolutionary distances. Okay. If you have a Mega software in your system, you can open it. Otherwise, we can download it from the browser. Go to your browser, search for Mega 11. Click on the link. You can see that there are some preferences needed. Based on your system, you can go to this. My system is Windows and it's 64-bit, so you can go to the download. And there is a user agreement. Accept it. And they need some basic details about you. And go to download. You can see that Mega is downloading here. I already have this software in my system, so I don't want to download it. And I'm straightly go to the Mega. Here is the Mega software. Here you can see that there are different types of options here. Before doing the phylogenetic tree, we need to import our sequence data. I already saved my sequence data here in a text file. From different 10 organisms. You guys can also do this. It will help you to easily access the data. Okay. So from here, go to the align option first. Click on it. And the first option, edit or build alignment. Click on it. And there will be a window here. You can go to the create a new alignment. So first of all, we need to align our data with a multiple sequence alignment. You can also download the multiple sequence alignment from an cluster W or cluster Omega, or you can do it here. Okay. Click OK. Here they are asking which type of data you are using here. Here I am using the protein data of insulin. So I choose protein. And you get an alignment explorer with a column, which there you can copy your query data. Here is our query data, the human insulin data from different 10 organisms. Copy and paste it in. Paste it here. So now our data is uploaded into the Mega. Before going to the alignment, just delete this empty row. And okay, first you have to select the all data. If you are not selecting the data, alignment will not be happen. So you can have two options. You can go to Control A, and also there are an option in edit select all. So selection will be like this. Okay. Now we can align using the alignment option. There are two options in alignment, align by cluster W and align by muscle. I am going to align by muscle. This is your alignment. Okay. Before closing the alignment explorer, we need to save our data because we need this data for our phylogenetic tree analysis. Okay. So here in data, there is a option export alignment. In three formats are here: Mega supports Mega format, FASTA format, and Nexus format. Now we are save our alignment file in Mega format. In data, there is an option of export alignment. Save your multiple sequence alignment in Mega format. Okay. I'm saving my data in the name of MSA. MSA stands for multiple sequence alignment. You need to give the name here in the input title of the data also and click OK. Now you can close this alignment explorer and go to the phylogeny. Phylogeny option will help you to get different types of trees. There are plenty of options here, but here in the demo session, we are going to the, in any method, I'm going to the neighbor joining method to create a phylogenetic tree. So you can choose phylogeny and click on the method based on your need. Here we are going to do neighbor joining method, second option. Click on the second option and choose your sequence alignment file. Here our sequence alignment file is MSA. Open it. Click OK. You will get your phylogenetic tree like this. And in Mega, you have different kinds of options here. You can change the font, color, and everything based on your need. Okay, that means the branch length can change from here, and the font, the distance. If here you can see the distance between each organisms. Okay, evolutionary distance. If you don't need that, you can hide it also. And also you can view in different formats from here, and you can change the color and font of the taxon names here. Like you can go to the red color, bold, 20, and then so we can see like this. And the tree style can change from here. The tree width can increase and decrease by using this option in the layout option. And there are some other options are here. From here, we can understand that how much the insulin protein are different in each organisms and how much they are related and divergent. Okay, here there is a difference in this tree. While we are already see in our PPT that there is a root in our tree, but here there is no root they are showing because maybe because these organisms of this protein from different organisms never have a common ancestor. That will be a reason for this too. Okay, so this is what phylogenetic tree using Mega. It's almost here. And you can also save this file if you are needed. I'm not going to save this. So do it by yourself and ask any doubt if you have. And now we are done Mega. And going back to our slides. Is there any doubt you can ask? And in conclusion, we are already explored the different sequence format, including FASTA and FASTQ, and discussed the significance in storing and analyzing biological data. We then delved into the phylogenetic tree. And you know that what is phylogenetic tree and their components, root, branches, and leaves, which illustrates the evolutionary relationship among the species. And phylogenetic trees are very crucial to understand the evolutionary history of organisms. Okay, aiding in applications like species identification, evolutionary biology studies, and drug discovery. By utilizing tools like MEGA, we can construct these trees to gain insights into the connections between species, enhancing our understanding of biodiversity and evolution. Okay, so I believe that you are all enjoy the session, and thank you for your attention. Is there any questions and doubts you can ask? Thank you all.